alaikum. I'm Asiya Sadiqa, and today the topic of my presentation for IBBC 2023 is genetic characterization of a family with autosomal recessive bipedal syndrome. So these are the outline of my presentation. First, I will discuss the introduction, then methodology, then results, conclusions, recommendations, and references. So first will be the introduction. It is an heritably clinically heterogeneous, pleiotropic and multisystem disorder. It has many phenotypes, showing many phenotypes characterized by the progressive retinal degeneration, polydactyly, obesity, renal irregularities to cognitive impairment. But it is also showing many other secondary medical conditions. It is a rare disease and estimated frequency is 0.7 in 100,000. And to date, 26 genes is reported to cause the BBS in diverse societies. And it exists in autosomal recessive pattern of inheritance, but also it is possibly inherited as an oligogenic trait. Second is methodology. First, what we do is that we take the informed consent form from the guardians of the patients. Then we do this blood sample collection. After that, what we do is the genomic DNA extraction, and it is using the same standard phenolchloroform method. After that, the whole exome sequencing and data analysis. And after that, what we do is that we design the primers for the pathogenic variants, and we run the PCR for the amplification of the specific sites. And then we perform this we do the PCR amplification, and then we perform the segregation analysis through Sanger sequencing to check the variant is segregating in a family or not. Research objective first was to investigate the genetic cause of BBS in a family from district cohort, and then second was the important objective was to develop the genotype-phenotype correlations among the family members. So these are the results. First, I'll show you the family pedigree. In this, all the members, the affected, normal parents, all are present in this family pedigree. Whole exome sequence data analysis. How we conclude this data is that we use different filters, like we use the genotyping, genomate frequency, coding effect, variant type. Like all of these filters are used, and at last we come to the conclusion. And these are all used to filter out the variant, the genetic or the causative variant that is causing the disease. So here we have found, after the using different filters, we have found our variant, that is a BBS7, and which is causing a non-frame shift deletion. So the gene is the BBS7. Mutation is at there is a sequence, and this whole exome sequence analysis show that the BBS gene is showing a 3 base pair deletion at the nucleotide position 580 to 582, and 3 base pair deletion removed an evolutionary conserved alanine codon 194, which most likely altered the secondary structure of the BBS7 protein. The genomate frequency is 0. Mutation tester is showing that is a disease causing. Franklin is showing it as a variant of uncertain significance. Versome is also showing it as an uncertain significance. This I have told you before that about the mutation tester. You can see here the results of the mutation tester. These are the results of the Franklin, which is showing it as a variant of uncertain significance. And this is the Versome, which is showing it as an uncertain significance. So now come towards the Sanger sequencing and segregation analysis. So as we have come to the conclusion that is a variant is in BBS7 which is like causing the disease, which is responsible after we do the whole exome sequencing of a abnormal family member. So now we see that segregation in the families is happening, or in what kind of situation is that. So let's come toward that. So first, there is a family member which is individual 2-1. Um, sequencing results showed it as an heterozygous, because here you can see there are two peaks. So it is showing it as in clearly showing it as an heterozygous, and we come towards the conclusion that is a carrier for this genetic variant. And the individual 2 was also at the same position. It is a genetic, it is also having two peaks, representing two peaks, and it is also heterozygous for the genetic variant. And then we'll come towards the individual 2-3. It is also showing the same situation is going on. It is heterozygous for the genetic variant. You can see clearly from the peaks that is showing double peaks. But the individual 2-4, you can see here in the position, at that position there is one peak, and there is no it is having the sequence, there's a GCT, you can see here, and it is having a single peak, and it is not, neither it is heterozygous, neither it is abnormal, it is a normal sequence. And the abnormal and sequence I'll show you on the next slide. The 3-4 individual is abnormal. 3-4 individual is also heterozygous at the same position, you can see here. And the individual 3-16 is also heterozygous for the same situation. And 3-3 is abnormal here. You can see here at the arrow that I'm showing you is that at this position there was a GCT that is deleted. This 3 codons letter, they are deleted. And also at 3-5, you can see here at this position there is a deletion occur. So now I'll come towards the conclusions and recommendations that the genotype-phenotype correlation established in the present study, it may improve the clinical diagnosis of affected individual for the better management and counseling, because it is very important to do it, because it is not only affecting one person or one individual, it is affecting the whole society. These are the differences. Thank you so much for your attention.